Hello guys. So today we are talking about our workshop is about the non-coding RNAs in general and the cysteine RNA in um, to be specific. So first you go to Google, you type in the HOMD genome browser. You click on the first link. So this is the home page. But what we are going to be focusing on is the origin. So yeah, it shows the different organism genomes, but we are going to be looking at the oral bacteria. Um, so here you can choose any organism, but it is better if you choose the Streptococcus gaudini. And then so it shows you the a description of that organism. So you click on the gene image map, select the chromosome 1, and then submit. So you have this page on. Okay. So in green you have the reverse trend. In blue, the forward trend. In gray is the introns. And as you go down, you'll see yellow, which means the ribosomal RNA gene, and in red you have the transfer RNA gene. And both of them are non-coding RNAs, of course. So as you go down, okay, you see this table, which explains you what everything is. So if you click on, for example, the ribosomal RNA sequence, so you have here the whole sequence of the ribosomal RNA, and actually some of them shows the protein sequence, but this one didn't. But I guess it is fine. We can maybe look at the transfer RNA. Okay, you see. So click on the one red. Okay, same thing. Anyway, so but we are going to be focusing on the cysteines. So type in Control F and then cysteine S. So click on that on the RNA cysteines 1. Might take a while, but okay. So here it is. So you have here the whole sequence. So you can select. Select the sequence. So Harrison is gonna continue with the presentation.
So now we have the 16S RNA nucleotide sequence. We can use it to actually blast and see if there's any other organisms which have it in common, which we're going to assume that there are because it's a highly conserved piece of DNA. So let's go back to the home page of the HOMD, the Human Oral Microbiome Database. Okay, and why don't you go to Dynamic Genome Blast, and you might have to wait a second for this to load. Okay, so your dynamic genome blast page should look like this, and it's different from a regular blast because it shows a phylogeny of all these different oral microbiome bacterial species and some archaea. So we're going to stick to bacteria right now because we know that the 16S rRNA is conserved in bacteria. So just select a few that you want to compare. And so I have 5 selected, and we're going to blast selected genomes. Okay, so we can paste that 16S rRNA genome sequence in here, and we want to make sure we're searching against genomic contigs, keep it standard, and of course DNA RNA query sequence type. Okay, now let's start blast. And what we're going to expect is that there's a very high degree of sequence similarity between this 16S rRNA gene and the genes that the other organisms have. Okay, great. So take a look at these e-value scores for these different genomes that we just blasted against. They're so low, it's 0, basically. The expect value is 0. That means that we can confirm that the 16S rDNA was highly conserved, so much so that it was exactly the same in the other organisms, essentially. Okay, and that's it.